Hello there, it's Mrs. Goebel, and today we are going to be looking at factors that can affect really the rate of photosynthesis. We are in Unit 5, which is the flow of energy. There are three objectives for today's lesson. The first is to explain how to measure the rate of photosynthesis. So that's kind of what we're going to talk about first. The second is to list all the factors that can affect the rate of photosynthesis, and finally to explain how each of those individually affects the parts of the photosynthesis reaction. So starting out here, you may be saying, okay, well, sure, photosynthesis happens, but how do scientists actually measure how well it's happening? Well, they do this with a rate. And what that means is the rate of photosynthesis is essentially just how quickly and how well a plant is able to take in that carbon dioxide and the water from the atmosphere and from soil and take those and turn them into glucose and oxygen. And the only way that you can really do this scientifically is to create a closed environment with the plant. And so then you'll either do CO2 usage or intake really from the closed environment, or you can look at oxygen that's produced. The other thing people do sometimes is take kind of like a leaf punch over time and look and see how much glucose or sugar is being produced. An example of this is over here to the right. This is a setup in a beaker with Elodea, which is an aquatic plant, so it's in water. You put a funnel around it, close off the top of it basically with a test tube, and if you see bubbles being made, that's oxygen. So that would be basically saying, hey, yes, photosynthesis is happening as oxygen is produced. So an easy way with that one is you just count the more bubbles that are made, the more oxygen is made. You can, like I said, do it kind of with leaf punches with glucose as well, or if the environment is closed with a plant, you can look and see how much CO2 actually gets used. That one's a little more complicated though. The reason, obviously, that we look at those individual factors is if you recall the equation for photosynthesis, carbon dioxide and water were both they're both reactants, and then our glucose and oxygen are both products. So you look and see, are any of the products made essentially with the oxygen? Okay, so there are a lot of factors that can actually affect the rate of photosynthesis, so how quickly it's happening. And there are 7 of them that we're going to go through, but I've bolded the first 3 because those are the ones people most commonly talk about. The other 4 do affect it, but the first 3, like I said, are kind of the most commonly cited. So the first here is intensity of light, and that's pretty much what it sounds like how intense the light is on the plant. And you could affect this by putting the light closer or further from the plant, and that's one easy way to do it. The seasons naturally do this just because of how much sunlight and how many hours of sunlight there is as well. But just basically it's what you would think. The more light you have, the better photosynthesis goes. And this makes sense because we have to excite the electrons in the photosystems, and that's pretty straightforward. You just have to excite the electrons. The problem here is if you get too much light, it's not going to make photosynthesis any better because it will eventually reach what's called a maximum rate, where again, actually adding more light is going to have no effect on photosynthesis. It's not going to make it go any better. So I've sort of said this a little bit already, but I do want to just make sure it's really clear. The light that is coming into the plant, remember, it's going to hit those photosystems in the light-dependent reaction, so those first set of reactions. The more light that is hitting those photosystems in the thylakoids, the more chance that you can have excitation, which is great. But once you start having too much excitation and too much light coming in, especially UV light, can actually damage these photosystems. So the little green dots, they're just representing our photosystems. Again, they can become damaged, but it's mostly because, if you remember, it's chlorophyll and proteins put together in the photosystems, and the proteins especially can be damaged, but the chlorophyll can as well. It's a compound. So too much light is a bad thing. It actually kind of breaks up or makes it so these don't work. Okay. Oh, this is doing weird things. Okay. So the next factor that affects how well photosynthesis works is temperature. And this kind of makes sense intuitively. You probably have some idea already about how well it's going to work. But essentially, if you have low temperatures, photosynthesis isn't going to work that well. So it's going to be pretty slow, or there may be none at all if it's too cold. At warm, or even what you think of as hot temperatures, that's going to be the range of best photosynthesis. But when it becomes too hot, so above 100 degrees usually, there's not going to be any photosynthesis because plants start to die. There are some plants that are adapted to these extreme temperatures, but they have special mechanisms to, kind of help keep things together. if we look at this this graph, you're going to see this looks pretty similar to some graphs we've seen before. So it doesn't work well at, a low temperature. It doesn't work well at the really high temperatures. And this looks so familiar because photosynthesis is going to do the same pattern that we saw with enzymes. Because the temperature, this curve we see, it looks just like one of those enzyme curves we learned a long time ago. Okay, so the deal here with the enzymes is in the Calvin cycle, if you recall, there were a lot of enzymes. And I didn't tell you the names of them, but they were the ones that were changing the sugars. So you started out with CO2 being added to RUBP, and then it was broken down into the 3-PGA and the G3P. There's so many threes, again, because they're 3 carbon sugars. So all of those, if you don't have the enzymes at the correct temperature, then they're not going to work. And so that's essentially why we see that pattern of, hey, it doesn't work too well at the high end, or doesn't work well at all at the high end and well at the low end. And that goes back to our enzymes. If it's too hot, the enzymes will denature. And remember, that just meant break down. So it doesn't work anymore. It unfolds. And it's just not going to be very active at low temperatures. So it's back to our enzymes. That's why it doesn't work. All right, so our next one here is carbon dioxide. And this makes sense because this was one of the reactants of photosynthesis. So they're going to have a direct relationship, which means when one increases, the other increases as well. So the more CO2 that's available for the plant, the more photosynthesis is going to happen quickly. And vice versa, if there's less carbon dioxide available, photosynthesis is not going to go as quickly. It will reach a maximum rate, again, just like we saw with temperature, where it cannot go any faster. So this is one concern people have if you've heard about global warming. And I think you guys can't really see this here, but the thought is, okay, CO2 is increasing in the atmosphere, and at some point, it's not going to help anymore. Plants won't be able to fix any more of the carbon. They won't be able to take it back out of the atmosphere. Okay, so you might be wondering, well, what happens if I change a couple of these? At any point, whichever factors in the least apply, so be it, how much CO2 is there, the light, whatever you're looking at, whichever one is in the least availability is going to have the greatest effect. So this graph shows, kind of if you have low CO2, low temperature, but a lot of light, what's going to happen? Well, you're going to be limited. It's better if you have more CO2, even if the temperature's lower. Best conditions, obviously, are a lot of CO2, a lot of temperature, and a lot of light, which is why, if you think about it, that's when plants are going to grow in the summer, right? It's nice and warm, the days are long, so this absolutely explains why we see plants in the summer, not so much in the winter. All right, so we did the first 3 that I said, those are the ones people most commonly cite, but there are quite a lot of other factors that affect photosynthesis, the first being the color of the light. So our chlorophyll and our other pigments, like the carotenoids, that we already talked about, are going to only absorb certain wavelengths of light, and we can see that very clearly in this graph down here. So chlorophyll A and B and the carotenoids, they're mostly absorbing, best absorbing in kind of the blue ranges, a little bit down in the orange and red, especially with the chlorophylls. But if you notice, most of them don't absorb green, which, again, that gets reflected, and that's why we see the leaves as green. So what you can actually do experimentally is you can try to either make it so the plant doesn't grow or make it so it grows even better. Because if you notice, again, most of what is being absorbed here is blue. So in a lot of greenhouses, they'll actually put kind of blue lights to best excite the chlorophyll A and B, just a lot of blue light. Conversely, if you gave the plant just a green light, if it's one that has these individual individual photosynthetic pigments, then you could actually kill the plant because it has nothing to absorb. So you can mess with the color of the light to kind of figure out what pigments are in there as well. All right, the next one seems pretty obvious. Is the availability of water. If you don't water a plant, it will die. If you give it too much, it's going to die too. Because the availability of water, again, water is one of our reactants in photosynthesis. What you may not realize, though, is the water is going to affect things in a little bit different way. And it really comes down to the leaves of the plants. The CO2 that I mentioned before, having a lot or a little bit, that just doesn't come down to how much is in the atmosphere. It comes down to how much is able to get into the plant. And the CO2 actually enters, and this shows water here, but CO2 goes in through these things called stomata. And here it's calling it stoma just because there's more than one. But an individual stomata, it's kind of like a little mouth almost for on the leaf. It opens, and when it opens, air is able to come in, and obviously air contains CO2. So CO2 is able to come into the leaf through the stomata. Well, if it's too dry, the plant doesn't have any water, it's going to close the stomata, and that means no more CO2 can come in. So photosynthesis will not happen when this is closed. There's nothing coming in. We're missing one of the reactants. Conversely, though, too much water in the soil causes the roots to rot. So if you have rotted roots, you're not going to get any more water, even if CO2 could come in. So essentially, without enough water in the right conditions, you're eliminating one of your reactants, and photosynthesis cannot happen. All right, the next one's a little bit weird. So humidity. You may not think about this, but a lot of tropical areas are very humid. And that's because if we think back to the stomata in just a second ago, they're going to open or close based on how much water is available. Well, if there is a lot of water in the air, which is basically what humidity is, there's not that much of a difference between how much water is in the air and how much water is inside the leaf. And remember that difference, anytime there's a difference between a separated area, that's a concentration gradient. So because there's not a big concentration gradient between the two, the plant isn't going to lose that much water when it opens up its stomata. And this means that less water will escape, and the stomata can actually be opened very wide. This allows more CO2 to get in, and photosynthesis is going to go faster. So that all seems pretty straightforward. The more water in the air, the more the plant can open the stomata, not worry about losing water, which again is why it has to close it back here if it's worried about losing water from the leaf out of the stomata. And then more CO2 gets in, photosynthesis is better. If humidity is low, the opposite is true. So Tennessee has some great conditions in the summer for plants to grow with high humidity. In case you're wondering how you would affect this inside, because obviously, outside the humidity is just controlled by the weather, but you can do a couple of things experimentally. People will use either peat moss, pebbles, or you can even put a brick under your houseplant. And when you water them, this will absorb things and then absorb water and then release water as well. So you can see they're showing that with a little humidity around the plant, and so that will help make it a more humid environment and photosynthesis to go better for your houseplants. Our final factor here is pH, and this is going to come back kind of to our enzymes again. So if you have soil that is too acidic or too basic, this can stop the photosynthesis of a plant. And this does come back to enzymes, and it's mostly one specific enzyme, but we have an enzyme called rubisco, which is ribulose 1,5-biphosphate carboxylase oxygenase, aka rubisco. Everybody calls it rubisco because that's an extremely long name. This is the enzyme during the Calvin cycle that is going to basically change our G3P, which was our sugar that would come out and be used to make glucose, back into our UBP, which kept our cycle going. So it's the enzyme that's going to be right over here. It does the ATP, ADP movement to make our 5-carbon sugars that we can use. Without rubisco, the Calvin cycle is going to just shut down, and nothing is going to continue. No more sugar is going to be made. Rubisco specifically is not going to work below 6 on our pH scale, and I just put this here to remind you that low numbers on the pH scale are acidic and high numbers are basic, or what they say is alkaline. So rubisco is only going to work within kind of this narrow range of basically 6 to 10, and if it's too acidic below that or too basic above that, this can actually shut down the photosynthesis of the plant. So just a reminder here, when we're looking at all of our steps in photosynthesis and where these things are happening, I did want to remind you, water goes into our light-dependent reactions, oxygen comes out, CO2 goes into the Calvin cycle, glucose comes out. A lot of these factors were affecting one or the other. Intensity of light is going to affect your light-dependent reactions. Water obviously will affect the light-dependent reactions as well as humidity. The color of the light as well is going to affect this stage, because if there's no wavelength of light to excite, this can't happen. Over here in the Calvin cycle side, we had pH is going to affect over here, temperature is going to affect over here, and then CO2 as well. So all of those would be affecting the Calvin cycle. Reminder, this does all happen inside your chloroplast, which is nice and green because of the chlorophyll. With that, we are at the end of the lesson. You need to write your complete summary.